All right. And we are back, this time looking at the lab manual. By now, I'm hoping that everybody went over to the DCC bookstore and picked up a copy of the lab manual. And as I am going through the lab manual and writing things down into it, you should be doing the same thing, right? You should be filling out your lab manual, filling in the same information that I am. There's going to be a couple parts with the lab that I'm going to skip over, and I'm going to expect you guys to complete that independently. Remember, filling out the pages of the lab manual is going to be important because when I give the lab exam, not only am I going to be asking for the answer sheet to the lab manual, but I'm also going to be asking for specific pages from these from this lab manual, and these pages will count as points towards the lab exam. All right, so the first lab is lab 1, using the compound light microscope. If we look at some of the objectives, it's going to be identifying some of the parts, describing the functions, looking at total magnification, using 3 different lenses, figure out how to focus on objects using the 3 different lenses, understanding orientation, so moving the slide in different directions and seeing how the image moves as we move the slide. And then the last thing that you would do if this class was to be performed in person, you would be preparing wet mount slides of your cheek cells. All right, so just a very brief introduction. This just describes what a microscope is, what its function is. The type of microscope that we use in this class is called a compound light microscope. And the why microscopy is considered an important technique. So what is a microscope? What is its function? Well, I'm not going to waste time writing all this in right now because the answer is right here, right? This is the answer. This one goes here. Microscope is an instrument used for viewing small objects. All right. Why is it important? Well, this one is right here. It's important in biology because a lot of what you're looking at is too small to be seen with the naked eye. We have to take a closer look. Things like cells, right, for example. So I would just copy those over. Holding a microscope, of course, it comes with an arm and a base. You put the handles on the arm, and then you always carry it with your other hand underneath the base. Cleaning the microscope lenses, we use these things. They're called Kim wipes. And the Kim wipes are designed in such a way that it doesn't scratch the lenses, right? All right, let's go over the parts. So the eyepiece is also called the ocular lens, and this is the part that you would be looking through. Now we have 2 ocular lenses. So this is a binocular compound microscope. Now, when I use the microscope, I guess this is as a result of growing up and through my high school experience and my college experience, I've always had the single ocular lens microscope. So lately, all the microscopes have 2 binocular lenses, but I still find myself closing one eye. I find that I could see better when I close one eye. It's difficult for me to use 2 eyes. It doesn't matter. Even if there's 2 lenses, you could close one eye and still use one. Now, this eyepiece, if you were to read these numbers here, you would see on the side that one of the numbers is a 10x. So this eyepiece, just by itself, the ocular lens magnifies the image so it appears 10 times bigger than it really does. And we haven't even gotten into the other lenses yet. So already the image is being magnified here. And then down here we have 4 lenses. But in our class, we only really pay attention to to 3 of them. 1, 2, and 3. This one over here, the biggest one, the fourth one, is called the oil immersion lens, and that one isn't really used in our class. It's mostly used in like microbiology classes. And the reason being is in order to use this lens, you have to put oil on the slide and create like an oil, almost like a tunnel between the lens and the slide. But with using oil, a lot of students forget to clean it up, and it makes a mess, and then it dries, and it's so for that reason we don't really use it in class. But we do use these 3. This smallest one has a red stripe on it. It's called the scanner lens. The middle one has a yellow stripe on it. That's called the low power lens. And then the other one, the bigger one down here, has a blue stripe on it, and that's called the high power lens. But collectively, we refer to these as the objective lenses. And how do we switch from one lens to another? Well, we turn this dial right here. This dial is called the rotating nose piece, and that allows us to switch between objective lenses. So we usually start with the scanning lens, the smallest lens. We find the image, we center it, we focus on it, and then we switch to the low power lens. Again, centering the image, focusing, before finally focusing using the high power lens. This area right here where the slide is going to be sitting on, this is called the stage, and we secure the slide with these clips. There's these special metal clips here that secure the slide, and these are called slide grips. Sometimes they're called stage clips. Down here is a light source, and what this does is it shoots beams of light through this diaphragm. This diaphragm is going to control the amount of light. We could open or close this diaphragm, which helps to control the light. And then there's a condenser lens in there that takes this light and kind of centers it on our specimen here. And then the light will continue through the lenses. All right, so the light source, the light is controlled by the diaphragm. This is the base of the microscope, the base. We could also control the intensity of the light bulb here. If we want to dim the light bulb, we have a little dimmer switch down here. So this is the light intensity adjustment knob. All it is a dimmer. There's 2 ways to focus the microscope. The first way would be to use this larger dial that's on the outside. Right, this one out here. This is called the coarse focus knob. When you are using the scanner lens or low power, you want to be focusing with this larger knob on the outside, which is the coarse focus knob. And what this knob does is it raises and lowers the stage. When we raise the stage and lower the stage, that's how we are focusing on our specimen here. Now, when we get to the high power lens, we want to focus with the inner knob here. The knob on the inside is a little bit different. All right, this is called the fine focus knob. And we use this when we're on high power. And the reason being is because when we're on high power, the slide is very, very close to this lens. So close, it almost looks like it's touching. And we don't want to move this stage up too quickly, because what can happen is we could either crack the glass on the lens or crack the glass on the slide. So as we're turning this, it takes several rotations of this knob even to move this stage the slightest bit. Okay, where if you turn this 1 or 2 times, you could see easily that this stage is going to be moving up and down. So this is a little bit more sensitive. It requires more turns to get it to move a millimeter compared to this one. All right. So there's greater distance between the low power lens and the scanner lens. So we can move this more freely up and down. So we would use this coarse adjustment knob. All right. So this is just a practice, right? This is what is this? This is just like a diagram, and we could practice labeling. So we could take care of this. This is our ocular. Here's our rotating nose piece. The rotating nose piece is how we switch between these objective lenses. Remember, there's 3 of them that we use. We could see 1, 2, 3 there. I can't really tell which one's which, but we're just going to collectively refer to them as the objective lenses for now, for this diagram. This is our stage. Here, now you can see the condenser lens in here. The condenser lens just kind of funnels all the light onto this specimen. Here's the diaphragm. Here's our light source. On-off switch. This is the coarse focus knob, and then the fine focus knob. And then over here we have our y-axis stage knob and x-axis stage knob. It's on the same. They're on the same device, right? So there's this little stem that comes down, and then the stem has 2 knobs on it, one here and then one here. All right. If we turn this one, this is the y-axis stage knob. So what that does is it manipulates the slide. The slide is going to be sitting within these stage clips here, right? And we could move the slide up and down. And when we're moving up and down, we're going up and down the y-axis. So we would turn this to move up and down. I can't really see yellow so good. Up and down. Where the other one, if we're moving along the x-axis, it would be moving left and right. So we can move the slide left and right by turning this x-axis stage knob. Left and right. All right, so by turning these 2 dials, we can move the slide left and right and up and down. The y-axis knob allows us to move up and down, and the x-axis knob allows us to move left and right. All right. We don't use our thumbs to move the slide anymore like we used to do in the older style. All right, so we could describe these parts. What is the eyepiece? This is the part you look through, right? And this has a magnification power of 10x. Let's add that because that's kind of important. All right. The rotating nose piece allows us to change between objective lenses. All right. Now, the objective lenses that we use, I don't know if you could tell, but this has a line going through it because we don't really use this lens at all. But we do use the scanning lens. The scanning lens is red, and it has the ability to magnify images 4 times. It has a magnifying power of 4x. It makes images appear 4 times larger than they really are. The low power lens is the yellow lens, and it has a magnifying power of 10x. So just like the eyepiece magnifies the image 10x, so does the low power lens. The high power objective lens, it has a blue stripe on it, and it has a magnifying power of 40x. So it will make images appear 40 times larger than they appear outside the lens. So the mechanical stage and the slide grips support the slide, right? The x-axis stage knob moves the slide along the y-axis, so that's up and down. Where the x-axis stage knob moves the slide along the x-axis, so that's going to be right and left. The light source directs a beam of light through the object, and the diaphragm controls the amount of light that reaches our specimen. We could reduce the amount of light reaching our specimen by turning this diaphragm and closing the hole. We could increase the amount of light by turning the diaphragm and opening the hole. And then we have those knobs for focusing. The outer one, the bigger one, is for focusing on scanner lens and low power. So the red lens, 4x, and the low power yellow lens, 10x. Where the inner one, the smaller knob, is the knob that we use for focusing on high power. All right. All right, very good. So how do we calculate total magnification of a microscope? Remember, we look through an eyepiece that has magnifying power, and then we look through objective lenses that have their own magnifying power. So when we're talking about magnification is making an image larger or appear larger. When we use a compound microscope, we're using two sets of lenses, the ocular lens, 10x, and an objective lens. Well, depending on which one, we have a 4x, a 10x, and a 40x. We have the scanner lens, the red one. We have a low power lens, the yellow one. And we have the high power lens, the blue one. So in order to calculate total magnification, we multiply the eyepiece towards whatever lens we're using. All right, so let's say we are using the scanner lens, the red scanner lens. Let's say we're using that one. Well, the red scanner lens by itself magnifies the image. This is the objective lens over here, 4 times. Where the eyepiece, the ocular lens, magnifies the image 10x. Let's erase that. All right. So we would just multiply 10x times 4x, and that gives us a total magnification of 40x. So when I'm looking at something in the microscope, right, and I'm using the scanner lens, the scanning lens, that image will appear 40 times larger than it does outside the microscope. When I'm using the low power lens, well, I'm still using the same eyepiece, so that doesn't change. But this lens has a power of 10x. So when I want to calculate the total magnification here, it's 10x times 10x. And that gives us a total magnification of 100x. So when I'm using the low power lens, that image will appear 100 times larger than it does outside the microscope. Lastly, if I switch to the high power lens, or most powerful lens, the eyepiece doesn't change, but this lens carries a magnification of 40x. When I multiply 10x times 40x, That increases the magnification to 400x. So this question now states, how many times larger does the object appear on high power compared to low power? How many times larger? Well, low power magnifies the image 100 times. High power magnifies the image 400 times. Well, 400 times is 4 times larger than 100 times, right? So it will appear 4 times larger when using high power compared to when you're using low power. All right, so in lab, you would be getting a slide, and on the slide, you would have a typed letter E from usually from like a newspaper or something, and then you would be asked to put the slide on this stage and try to focus, and you would want to use your X and Y adjustment knobs to get the slide to be centered and that letter E right over the light, right over this beam of light would be coming through, and that you would be asked to just put this slide in the stage clips here, and then use these two knobs to position the letter E so it appears right in the middle here. So that's what you would be doing, and then you would be raising the stage up and down as you turn this focus knob, the coarse focus knob, the one on the outside, the larger one. And then you would adjust your oculars so it's comfortable to your face. And then as you're looking through, you would continue to focus until the image becomes visible. Now, what you would do here in this case is I would ask you, draw the image as it appears on the slide. Well, we just saw this picture of the slide here, or a drawing of the slide up here. So this is what I would want you to draw here. This is how it looks on the slide. It's right side up, right? It's normal looking. It's small. So you would copy the image just from how it looks on the slide, and you would probably draw something like this, right? And as you use the scanner lens and focus on it, what happens is a couple of things, all right? Now, what happens is if I put this on the microscope like this, the first thing that happens is the image becomes reversed. So it'll get flipped around like this. But then it also gets inverted. So it gets flipped twice inside the microscope as it goes through the two lenses. So then it'll look like this. So it goes on the microscope looking like this, but inside the microscope, it gets flipped twice, it gets reversed, and then it gets inverted. And not only that, but also as I look in the microscope, the image becomes magnified. So it'll appear larger. So the second drawing is asking you to draw the image as it appears in the microscope. And you would then draw something like that. All right, showing that it is indeed flipped 2 times. And you would also draw it bigger because it's being magnified 40 times when we're using scanner. So this is a multiple choice question. How is the image of the letter E under the microscope? Well, this is the one right here. This is the choice we would go with, this one. This is what it would look like if we put it on looking like that. So 3 changes. The image is reversed, it's flipped upside down, and then it's bigger. As you use the microscope, you would notice if you turn the x-axis knob and the slide goes to the right, what happens to the image is the image will move to the left. And if you then use the y-axis knob and you move the image down toward you, the image will move away from you. So, in other words, if you move the slide in one direction, the image will appear to move in the opposite direction. So, after you're focused on the red scanner lens and your letter E is centered and focused, you would then be directed to change the lenses by turning the rotating nose piece, this time to the yellow colored low power lens. The low power lens, remember, has a magnifying power of 10x. All right, so when you do that, you would turn the rotating nose piece, it would click in, you would look through the oculars, it would be fuzzy. You would have to turn the coarse focus knob until it becomes clear, and then you would use your X and Y adjustment knobs to center it again. Now, what would happen is you would then be asked to draw what you see again. Now, what happens is the letter E is getting bigger. In this case, it almost fills up the entire field of view here. We could still fit it in, but it takes up almost the entire space. It would look something like this. Many of you would be drawing something that looks like that, right? It's a little difficult to do using a stylus on the computer. You would probably keep it within the lines. I went over the lines, but yeah, it would be nice and thick and big. You could still see that it's a letter E. And then you would draw it like that. This is where you would record the observation. You would make sure it's centered and focused, and then you would be ready to turn the rotating nose piece one more time to the blue powered lens, which is the high power lens. Remember, the blue power lens magnifies the image 40 times, right? So here's the thing. You really want to make sure what you're looking at is centered before you go to the high power lens, because if you don't center the image, there's a chance you could be zooming in on this area right here or this area right here. And then if you zoom in on that on high power lens, you wouldn't see anything. So what I recommend to the students is take this area and put it in the center before you go to high power. This way we know for sure that we can see something. And when you switch over to high power, what you would see if you moved in on that is, just like a big smear of ink, really. Maybe it will go out this way too a little bit. So you'd really be zoomed in on the ink. And this question is even asking, can you still see the entire E? And the answer is no. I'm only zoomed in on a certain part, right? It's not recognizable as a letter E anymore. It's just like I'm zoomed in on ink. So why is that? Because, well, this high power lens has a magnifying power of 40x. And when I combine it with the eyepiece that has a magnifying power of 10x, this image is looking 400 times bigger than it does outside the microscope. This one is 100 times bigger. So the high power view is 4 times bigger than the low power view was. This brings us to what's called field of view. When you look through the microscope, your field of view comes in the form of a circle, right? You see if there's nothing on the microscope, if you just turn the microscope on, don't put anything on it, and look inside the hole, you're going to see a big circular area of light, right? This area of light can be measured in a diameter. There's an actual distance that you could measure from here to here, all right? And this is called the field of view diameter. Now, in class, what you can do is you could take a clear ruler and put the ruler in there and measure the amount of millimeters going across for each lens. Remember, each lens is going to be different because they have different strengths. But if you were to do that, you would find that in the red scanning lens, if you put the ruler up there and measured millimeters, um, the total magnification of the red scanning lens, which is 4x when combined with the eyepiece, is 40x, right? You would measure that this lens shows you about 4.5 millimeters. So it would look like this. This is from here to here is 1 centimeter, right? But 1 centimeter is divided into 10 millimeters. Every space between the lines is a millimeter. So this is a millimeter. This is 2 millimeters, 3 millimeters, 4 millimeters. And if you were on the actual microscope, you would see that it would go about your field of view. You would be somewhere in the middle of this fifth millimeter, and you would estimate that this is about 0.5. So the field of view when using the scanner lens, the red lens, the smallest lens is 4.5 millimeters. Now, when you zoom in and go to the yellow low power lens, this low power lens has a total magnification of 100x. This is indicated by this circle here. And you would estimate that you could see pretty close to 2 millimeters, maybe a little bit shy of 2 millimeters. So you would estimate that. Most students say 2 millimeters. I can see 2 millimeters. But it's a little bit shy. It's actually 1.8 millimeters. And then when you go to high power, when you turn the rotating nose piece to the high power lens, this lens has a magnifying power of 40x, but when combined with the eyepiece, that becomes 400x. Well, you could see that's illustrated by this circle here. And you can't even see an entire millimeter. In fact, it's not even half of a millimeter. It's actually 0.45 millimeters. So the distance across in millimeters is 0.45. So it's just short of 0.5, which is half of a millimeter. So as magnification is increasing, the amount of space that we're looking at is decreasing, right? As the lenses get stronger, we're really zooming in on objects. And the amount of space that we're seeing becomes smaller because we're getting closer to it, right? So the total magnification of low power lens is more or less than the total magnification of the scanning lens. Well, the total magnification of low power lens is more because 100x is more than 40x. However, the field of view of the low power lens is smaller than the field of view of the scanning lens because the field of view of the low power lens is 1.8 millimeters. But on the scanning lens, the field of view is 4.5 millimeters. We can see this much space because the scanner lens is not very powerful. So we're really taking a step back and looking at, let's say we're looking at that letter E. That letter E would be small here, right? And as we go to the low power view, then that letter E becomes bigger because we're getting in closer. So what is the relationship between magnification and field diameter? Well, in this case, we have what's called an inverse relationship. That means as one goes up, the other one goes down. So as total magnification increases, field of view diameters decrease. So it's in this part of the lab that I would be asking you to prepare a wet mount slide, right? When you prepare a wet mount slide, what you do is I'm probably going to add a YouTube video of somebody actually doing this so you can see how to do it. But what you would do is you would just take a slide, put it on a paper towel. You would add a couple drops of some type of stain. A lot of times when we're looking at epithelial cells that we collect from our cheeks, we would use either iodine or a different type of stain called methylene blue. And the reason why we want to add a stain to our cells is because the cells are clear, they're colorless. We wouldn't really be able to see much. But the cell membrane and the nucleus, they do a pretty good job of absorbing the stains. So then those structures become visible. All right, so then you would take a toothpick and kind of rub it around the inside of your cheek, and then take it out and gently swirl the toothpick through the stain on the slide, hopefully collecting some cells from the toothpick as you harvest the cells from the inside of your mouth. You would place a cover slip on top of that, and then you would put it on the stage, securing it on the stage with the stage clips, right? And then going through the steps of, raising it up, using your X and Y adjustment knobs to move it back and forth. And then you would draw what you see here. So this is what I'm going to leave you with here. I would like you to draw these types of cells that we're looking at are called simple, squamous, epithelial cells. Epithelial cells are the types of cells that usually line body cavities, right? Your oral cavity is inside the mouth, so epithelial cells will be there. Squamous refers to the shape. So epithelial cells usually come in three different shapes. They're either cuboidal, shaped like cubes. They're columnar, so shaped like columns, long and skinny. Or they're round and plate-like. And in this case, these are called squamous cells. So they're roundish and plate-like. So this is the kind of shape that we're looking for. This is typical of what's coming out of your cheeks. Simple means that they're not really stacked one on top of another, that they're arranged in singular levels. All right. So simple squamous epithelial cells are the cells that we're trying to harvest from our mouth. And if we go to the lab atlas, do I have that open somewhere? I do. The lab atlas, remember, is an important tool that's provided to you. It's on Blackboard, and it's a good place to come and review all the different material. And this goes over the parts of the microscope, right? The steps on how to focus on something using the microscope. But this is what I want to really focus on here. Now, these are the cells. So this would be using the scanner lens. Now I could see a nice cell here. This cell, I could see a dark dot in the middle. That would be the nucleus. And then this outer edge is the cell membrane. And then everything in between, we could just call that cytoplasm. So this is what we would be looking for. We're using the red scanner lens. These cells are being magnified 40 times bigger than they appear. You couldn't be able to see this with your eyes. They're too small. But from here, we would want to zoom in, right? So we're using that 4x lens. Total magnification when we add the eyepiece in is 40x. And this field of view, the distance across is 4.5 millimeters. But millimeters are actually too big that we use a different unit. It's called micrometers, which is 1,000th of a millimeter. So there's 1,000 micrometers inside 1 millimeter. And if we establish that the distance across is 4.5 millimeters, well, we could also say that the distance across is 4,500 micrometers. So you would draw this in the first circle. That's a cell. That's a cell. That's a cell. These are all cells. Now, as we go to low power, we can see the cells are starting to get bigger because the low power objective lens is 10x, giving us a total magnification of 100x. Remember, the distance that we measured with our rulers was 1.8 millimeters, but when we convert that to micrometers, remembering the fact that every millimeter has 1,000 micrometers in it, that changes the field diameter to 1,800 across. There's a cell. There's a cell. There's a cell. This is a great cell because I could clearly see the nucleus there. I could clearly see the cell membrane. And all the light purple, all that fluid inside would be the cytoplasm. Here's another good one here. I'm blocking this one. And then lastly, we would go to the high power view. All right. So the high power view, the blue lens has a 40x magnification or 400x magnification. Remember, with millimeters, it was 0.45 millimeters. So when we convert that to micrometers, that becomes 450 micrometers. And why is that useful? When I know the distance across, all right, my field of view, I could use that distance to help estimate the size of the cell. So if it's 450 micrometers all the way across, I would just have to ask myself, how many cells can I fit across in a single line if I could do that? So I would use some type of measuring device. In this case, I'm going to use my fingers. And I would estimate that maybe 9 cells. If I could fit 9 cells across, I would take my known field diameter of 450 and divide it by 9. And that would give me what? 50 micrometers. So I would estimate that the size of this cell is about 50 micrometers by using my known field diameter and estimating how many of these cells I could fit across. It's kind of like estimating the size of your car by knowing the length of your garage, right? If you have a garage that's 20 feet across and you put your car in there and you estimate that you could fit 4 cars in this space, well, then I could kind of figure out the width of my car. I'll go 20 feet divided by 4, right? Which is 5 feet. You would estimate that the width of your car is 5 feet. We're doing the same thing here. This is kind of like the size of a room. But we're not using feet. We're using different units. We're using microscopic units of measurement called micrometers, right? So we know that it's 450 across, micrometers across. We've established that. That's never going to change when we're on high power. Now we just have to figure out the size of this, knowing the distance across. Here we see the nucleus, the cytoplasm, the cell membrane. All right, so go back to the lab here. You're going to do your cell drawing as it appears on scanner view, and then again on low power. And the last one you're going to do is the high power view. Now, remember, when you do this, when you draw it, as we just saw from the images, you're going to label the cell, you're going to label the nucleus, label the cell membrane, label the cytoplasm. This is important. You're going to fill in your magnifications, the eyepiece, the objective lens, and this is going to be the high power objective lens, the total. Now, what I want you to do here is estimate the size of the cell using the field diameter, right? Remember, the field diameter has already been established. The distance across here is 450 micrometers. So you don't have to draw that on here. I'm just showing you. And if I was to draw something that I'm looking at under the microscope, let's say I drew this thing here. whatever this is. Let's say that this is something I'm looking at under the microscope. If I wanted to estimate the size of this, I would ask myself, how many of these things could I fit across? And, it doesn't have to be perfect, right? It's just an estimation. So I would say, I could probably fit another one over here, right? That's one, two, and then I could fit another one over here, three, and then another one over here, four. And then maybe a half of one over here, 0.5. Well, what is 450 divided by 4.5? Well, that's pretty easy math, isn't it? In this case, it is 100. So I would estimate that the size of this is 100 micrometers. And I'm estimating the size of that using my known field diameter. All right. So that's how you do that. So do your best to do that. Don't add all these things that I'm adding in mind. You're just going to use the drawing that's from the lab atlas, and do the best to draw it. All right. And that's the last thing that you're going to have to do. Remember, you have to fill out all the pages because you're not going to know which pages I'm going to be asking for to be submitted with the lab exam. So make sure you have them all done. All right. So this is the introduction to the microscope. And yeah, I mean, it would be nice if we had real microscopes in front of us using them. But this is the best that we got. All right. So, if you have any questions, feel free to email me. But I think this is pretty much straightforward. And until next time.